The topics of the screencast include membrane proteins and cell junctions, which you will find in Chapter 3 of your textbook. We're going to start first with membrane proteins. Your learning objectives are to explain the roles of membrane proteins as follows: as ion channels and carrier proteins, as receptors, as enzymes, cell identity markers, sites of attachment, and the role of membrane proteins in cell-to-cell joining. Transmembrane proteins play critical roles in the movement of substances across the plasma membrane. Some carrier proteins can act as ion channels, as shown here on the left. An ion channel is basically a little tunnel that allows for substances to cross from one side of the plasma membrane to the other. Typically, the portion forming the borders of the tunnel are hydrophilic, and they allow for hydrophilic substances, therefore, to pass through. Note that sometimes these ion channels can be gated, meaning closed at sometimes and open at other times. In addition to serving as ion channels, transmembrane proteins can also be carrier proteins, where they will bind the substance on one side, change shape, and release it on the other. So they're different from ion channels. Ion channels are like tunnels. You could think of carrier proteins as actually grabbing, pulling, and releasing the substance onto the other side. In this example, the carrier protein has to be energized by ATP in order to change shape and move that substance across the plasma membrane. But I do want to note that not all carrier proteins require ATP. Some do, some do not. This is just another illustration of the action of a carrier protein. You can see here a substance entering the carrier protein and then binding to that carrier protein changing shape and releasing that substance on the other side of the plasma membrane. This conformational change in shape sometimes requires ATP, sometimes does not. If it requires ATP, then we refer to these carrier proteins as pumps. Transmembrane proteins can act as receptors. Receptors have a binding site on the extracellular face of the plasma membrane. And when a chemical messenger, such as a hormone or a cytokine, comes along, it binds to the receptor. This binding changes the shape of the protein, which then leads to changes in chemical reactions occurring inside the cell. This is called cell signal transduction. Recall that enzymes are proteins, and some transmembrane proteins can act as enzymes, either on the intracellular face or the extracellular face of the plasma membrane. And in fact, you can have several enzymes adjacent to one another, or several of these transmembrane proteins acting as enzymes adjacent to one another, and they can actually catalyze sequential enzymatic reactions in a metabolic pathway. So for example, here we have a substrate, and it is a substrate in a chemical reaction, or this is the product, and then this product becomes a substrate in a second chemical reaction, forming this product, and then you could have additional enzymes that would then take this product, and it would be a substrate in an additional chemical reaction, and so forth and so on. So you can have teams of adjacent enzymes in the plasma membrane catalyzing sequential chemical reactions in a metabolic pathway. Membrane proteins can also serve as cell-to-cell recognition or identity markers. Recall that some proteins have a carbohydrate component or moiety, and they are part of the glycocalyx. Remember, the glycocalyx is the sugary coating on the external face of the plasma membrane. Well, do you ever wonder how our white blood cells distinguish between our self cells and foreign cells? Well, that is where the glycocalyx comes in. These glycoproteins serve as little "hello, my name is" stickers and allow immune cells to identify cells as belonging to the body. These are also referred to as self-antigens. And if the white blood cell does not recognize the cell's antigen, it would attack it. So this is how your immune system is able to distinguish cells that belong to the body versus cells that are foreign. It's the arrangement of these glycoproteins in the glycocalyx. Of course, the arrangement of these proteins in the glycocalyx is going to be determined by your DNA. Membrane proteins also attach to the cytoskeleton on the internal face or intracellular face of the plasma membrane and to extracellular matrix such as proteins on the extracellular face. These attachments help anchor the membrane proteins to fixed positions, which can be important in maintaining the shape of the cell, can play a role in cell movement, etc. Also, cells often do not grow normally or survive unless they're mechanically linked to extracellular material. So these attachments to the extracellular matrix are very important. Cells often interact with their neighbors, and the proteins on the extracellular surface of their membranes can interact with the proteins on the extracellular membrane of adjacent cells, forming multiple types of what are called intercellular junctions. Some of these membrane proteins form what are called cell adhesion molecules that can provide temporary binding sites for guiding other cells during cell migration, as well as other cell-to-cell interactions. Before ending our discussion on the functions of membrane proteins, I just want to note that both transmembrane proteins and peripheral proteins can act as receptors, enzymes, participate in cell-to-cell recognition, attach to the extracellular matrix and the cytoskeleton, as well as participate in cell-to-cell joining. However, only transmembrane proteins can serve as ion channels and protein carriers because, of course, they have to span the entire width of the plasma membrane. We will now turn our attention to cell junctions. Your learning objectives are to describe the following cell junctions: tight junctions, desmosomes, and gap junctions. Cells often have specialized structures in their plasma membranes that allow them to interact with neighboring cells. Sometimes they're physically joined together, and other times they allow the cells to communicate with one another. There are three junctions you ought to be familiar with. They are tight junctions, desmosomes, and gap junctions. Tight junctions are impermeable junctions. They do not allow substances to move down between adjacent cells. If we look at a tight junction, what we find is that the proteins on the adjacent plasma membranes have fused together. We would find tight junctions in parts of the body where we don't want substances to move down between cells, such as the digestive tract. So here we have the surface of the small intestine. We have digestive enzymes here digesting consumed food. Well, we wouldn't want those digestive enzymes to move down between the cells because they'd start digesting, literally digesting the cells themselves. There are also other parts of the body where we don't want substances moving between cells. Another location would be the capillaries of the central nervous system. There we have the blood-brain barrier that prevents substances from moving from the blood into the central nervous system, the brain specifically, rather. And there, the cells that line the capillaries of the brain contain tight junctions. Desmosomes are anchoring junctions. They consist of various proteins that anchor the plasma membrane of one cell to the other. The function of desmosomes is to resist cells from being pulled apart from one another. Typically, they're found in tissues that receive a lot of mechanical stress, such as your skin and cardiac muscle cells of the heart. As cardiac muscle cells beat, or contract, rather, they would tend to pull away from one another. The desmosomes anchor the cardiac muscle cells to one another so that this does not happen. Gap junctions are best described as communicating junctions. They consist of transmembrane proteins on adjacent cell plasma membranes, and they allow substances to pass between the cells. Those substances can be ions, sugars, and other molecules. And though it may sound strange, this is how one cell can communicate with another cell or send a message to its adjacent cell. You'll find gap junctions in cells such as cardiac muscle cells, and it's one way that the cardiac muscle cells can coordinate contraction as ions pass from one cardiac muscle cell to the next. Now let's review the learning objectives of this screencast. We started with membrane proteins. You must explain membrane proteins as ion channels and carrier proteins, receptors, enzymes as cell identity markers, sites of attachment, and the role they play in cell-to-cell joining. We then concluded by looking at the three types of cell junctions: tight junctions, desmosomes, and gap junctions. Be sure you can describe each.